Yes, good evening to all the participants. So, welcome to the day three session on computer aided drug and vaccine design. So, today we'll be discussing on drug likeness properties, drug ADMET properties. So, before going to step on to day three session, the left out part on yesterday, like yesterday, I have taken you to the Swiss modeler, isn't it? In the Swiss modeler, I mentioned that I'll be showing you the results on today. Okay, so if you see the Swiss modeler, I have received the results on yesterday after 10 minutes, and these are the results. Okay, so that is, I have created a project called Project One, and I have received the results in email. Okay, so as I mentioned you earlier, the Swiss model works on homology modeling. Okay, this is one of the method of protein structure prediction. If you want to build a protein structure for the structure is not present in databases, we have to feed the target sequence of a protein. Whatever FASTA format of a protein sequence, if you feed in Swiss model website, okay, the Swiss model is present in XPASI web server. So I just uploaded the sequence in FASTA format. And if I click submit like build model, I'm getting these two models, model 2 and model 1. To download, to check which is the best model, first we can check even with respect to sequence identity. Then sequence identity, we can able to see how many number of amino acids of target sequence. That is, when I worked on yesterday, you remember, I have taken mouse trap1. So trap1 is one of the target receptor of cancer, I told you. So for the mice alone, we don't have the structure, but for human, we have the structure. So when we try to build a template PDB structure, I got sequence identity 88.49 for model 2, but for model 1, I got 78.53, which is less. So which is most important? Like if you want to select a PDB, of course, the sequence, target sequence and template PDB protein sequence should have more identity. Okay, so I suggest you to download model 2 when compared to model 1. This is how we can decide, even if we get two best models, which is the best model, we can choose based on the sequence identity. To download the file, click this download file and download this PDB format. Okay, so this is how we can able to download the model structure of a protein. So I've created a folder called CADVD. So I'm just saving this model as model 2. If you want to open this model 2, open it using BioVia Discovery Studio Visualizer. Like however I opened and showed you on yesterday, similarly we can try like that. Okay. And to assess the stereochemical quality of a protein, to assess the stereochemical quality of a protein, I told you we can use Ramachandran plot. So the Ramachandran plot can be checked using by clicking the structure assessment. If you click the structure assessment, you can see the Ramachandran plot. We have four types of Ramachandran plot. The general will have all the amino acids of the protein. You know, the protein sequence I have taken is myostrapin, which is around 736 amino acid. So all the 736 amino acids are scattered in this 2D graph, that is psi versus phi. Okay, and if you see these amino acids are not lying in the dark green regions. So this amino acid, so these are unfavored regions. These are unfavored regions which we don't want. Okay, so if you see the favored regions of Ramachandran, it comes around 95.45. So if you whether this protein can be used further for docking or simulation, this can be decided based on the Ramachandran favored regions. The Ramachandran favored regions, if it is above 80, 80 and above, we can choose it for these kind of computer drug design applications. So in our case, this is above 80, so we can use it. So this is how we have to assess even the protein quality after developing the protein model using the Swiss model. So developing the model, assessing the protein quality, everything is available in the Swiss model server, which works on homology modeling principle of protein structure prediction. Okay, and related to the feedback of on day two, I got a good kind of a response from most of the participants that you appreciated the way, but you find some kind of lag in audio and video quality. Actually, if I send you this kind of when I say you might have notified my recorded videos. You check whether whatever videos, the recorded videos I have sent, you have finding lagging in audio or video. So that is based on your internet bandwidth. If your internet frequency is less in your specified region, then obviously you find the lag in audio and quality, audio and video quality. So I suggest if you find such a kind of a problem, whatever video I'm sending you, okay, through I'm sharing it to you, you better watch that kind of a video so that you whatever you missed in between, okay, in between the internet bandwidth, so that you can able to compensate. Okay, no doubt I am sending all these videos, isn't it? Two videos I've already shared to you. Anyone face problems with receiving this video to your email ID? Anyone didn't receive the video? If not, you just message or you just unmute and speak, because I like to know, because many of them also given that more than 90% of participants have given the feedback that there is no problem even with the video and audio, but for 10% of participants they face the problem. So for the 10% of participants, I was just mentioning you that it might be your internet band, because 90% of participants accepted, isn't it? Then you too have to accept. That's the factor. So better check the recorded video. If you find lag in audio and video, try to compensate. Okay. So now let me go ahead with a recap of the last session topics. I just come across with whatever we left out. Like I told you that I assured you that I'll be showing you the output of Swiss modeler. Now I showed you. Now let me come across the recap of last session topic. That is day two session. Recap of last session topic. Even I found many participants appreciated this pedagogy, that is recapping and going further with the next session topics. Can we go ahead? What exactly did we discuss in the last session? If you are not recapping, we are continuously moving like a goods train. We don't know where exactly. So, okay, can come across.
So we started with clinical research phases and we saw NDA, INDA,
Hmm. So very good. We have
and
we
and
have this.
deep dive into databases, which is
Hmm.
open for practice. Around 12 to 13 databases, he told us.
Good. Good. Very good interaction from Madhu Krishna. See, actually, even I got one feedback from a participant, okay, a faculty participant, that whatever we missed, like whatever I showed you in the form of a slides, she want me to show in the form of skill, like showing in the browser how to work with the database and tool. Definitely, we'll be working that. Okay, whenever we find time we can work on that. I believe that even a 1 hour session, I'm extending up to a minimum of 1 hour, 5 minutes, something like that, isn't it? So even under 1 hour, 5 minutes, I can't able to complete. I'm making a very compact to complete it. So even the output, the output of yesterday's session, I'm showing it today. Okay, so it happens obviously if the output generates late in web server. Okay, so whatever you feel something one or two is missing, like what one or two databases illustration is missing, that you will be seeing this, or you can see the compensation in the forthcoming sessions. So try to attend even the forthcoming sessions. A good recap from Madhu Krishna. But only one participant has recapped in the form. But how about the others? No one has even typed the message. Message me like what exactly did we recap? We have discussed on methods of protein structure prediction, isn't it? Now I can ask you the questions. What are the three methods of protein structure prediction? Protein structure prediction. Did I discuss three methods of protein structure prediction? What are the three methods? And the three methods I showed you in XPassive. Very good. From Vishnu, you came across with two methods, threading and ab initio. One is missing. What is that? Threading is one of the method. Homology modeling. Very good. So, what are the three methods now? We just recap: homology modeling, threading, and ab initio. Okay. And my next question is how to assess stereochemical quality of a protein. Stereochemical quality of a modeled protein. So if you don't have a protein structure, you're modeling, isn't it? How to assess? Yes, very good. From Ramachandran plot. Ramachandran plot is a plot uh, 5 versus 5 versus what? So it is a plot 5 versus psi. Exactly. Okay. So we have discussed about this. Okay. Now let me come across on today's session on ADME properties. Okay. So I request Adhikari can go ahead by sharing your presentation.
Thank you, sir, for this opportunity. I am going to share my slides. Uh, 1 minute. Sir, it is visible.
Yes, it's visible.
Okay, sir. I am starting. Hello, everyone. Good evening. I am Kaustav Adhikari. Let's introduce me. I am the PhD student of Dr. P.K. Praveen Kumar. I am today. I am going to discuss about the chapter 3, the drug likeliness properties and the drug databases, drug admit properties. Actually, uh, in this session we will go with the interactive sessions. You can all the participants can interact with me in this session via unmute their mic, and also they can ask me question when they have any doubt and anything. I will definitely answer you. Okay, let's start this session. So, what about this today's session is contents like introduction to the drug likeliness, why the drug likeliness matters, core principles of the drug likeliness, rule-based filters for the drug likeliness, admin profiles, and various softwares for the prediction. These contents will provide this to the session, and after that, in this session, we will go through the various softwares for the prediction of drug likeliness. So we move on to the first session, introduction to the drug likeliness. Uh, let's see. Uh, anyone can tell me if we have a mango tree and we aqueous extract we collect from the mango tree. Can we and we have a extract like mangiva and mangiferin, like this type of abstract extract. Can we state them that is a drug or not? Can anyone tell me by unmute their mic? Anyone?
You can pitch the question once more, and also
Okay,
you
sir.
click this hide instead of
Okay,
hide.
sir.
Click that hide.
Okay, sir. Now we can see the presentation.
Yes. Yes.
Yes, sir. Can you anyone tell me if in one mango tree we are collecting from the one aqueous extract collecting from the mango tree, and we get some aqueous extract like mangiferin and others there, we can we state that this mangiferin is a drug or not? Can we state that? How we can state it is a drug or not?
Only when it has a therapeutic effect.
Yes, very good. When it is a various therapeutic effect, we can state it a drug. But only that is that we can refer it's a drug. It is not. Actually, when we there is drug likeliness is a property. When we can refer this is a drug or not, that is the main thing in this topic. We can drug likeliness is referred to the qualitative assessment of whether a chemical compound has a properties consistent with an orally active, safe, and effective drug, which means that there one chemical compound that is have a orally that is an orally active compound. That means we can take it as a orally and take it after taking it will affect some showing safe effect and it effect on the target, mainly effect on the target. It does not guarantee that activity or clinical success. Instead, it helps filter out the compound's unfavorable ADME toxicity, ADME or toxicity profile in the drug discovery. Like when we are going to present a drug or in anything, chemical compound, it is chemically succeed or instead of this, it is helps to filter out the compounds with unfavorable ADME toxicities. ADME toxicities, we are coming about this topic after some time, but at first we have to know that drug likeness is a property that ensures that it is a drug or not by a qualitative assessment of this chemical compound. So chemical compound, this drug likeness integrates some physicochemical properties, ADME parameters, structural alerts, synthetic feasibility, and computational scoring. These are integrating the drug likeliness properties. So that we come from the drug likeliness. What is the importance of this drug likeliness? This is drug likeliness reduces the attrition in drug development. Means the decay of the property of the drug is also decayed by absorption, metabolism. So that it reduces the attritions in drug development. It also guides the rational drug design and improves the compound library selection and enhance the success rate of lead optimization. Can you go through the what is the lead? From the first session, we know about what is lead. Can anyone tell me what is lead? Please unmute and tell me what is lead. Anyone? Lead is a chemically in the first session we know about the lead. Okay, so what is lead? Lead is a that is a type of chemical compound that have some potential to effect in the effect like a drug. Lead is not like a drug. Lead is not the effect drug, but it's like a drug that has a potential to be a drug. Okay, like it is in a layman language, it has a potential to be a drug. So it will be the enhance the success rate of lead optimization. Lead have to be optimized, after that it will be a drug. We call it that after that we call it a drug. So in enhance the success rate of the lead optimization, it is drug likeness property is important. So we are coming to the physiochemical properties. At first, what is the key physiochemical properties of the of a drug? Molecular weight. Can you tell me molecular weight? Why it is important? Molecular weight. Anyone? Why molecular weight is important? Ah, yes, permeability. Good, good, Vishnu. And anything, any answer? Okay. Molecular weight is important because of permeability. Because we know that the whole the scenario is going through the in a type of a whole the scenario is going through the type of a body, human body. We are talking about the human body or any animal's body. We incorporate the drug. So for incorporating the drug, we need to at first we need to know what is the what how do the drug reach to the blood. At first, we liberated the drug with liberation of the drug at in that LADME properties. At first, we liberation the drug. We after liberation the drug, it is taken by orally or intravenous. If it taken by orally, it will come to the GI tract. After getting to the GI tract, it goes through the stomach, then comes to the intestine. By through the liver, this comes to the intestine. After that coming in intestine, there will be the absorption, and after absorption it went to the blood vessels and came into the blood, and blood circulates this drug to the effective target point. It is the main procedure, but in this procedure there is some problem, because in the time of absorption it comes to the GI tract to the blood via some uh, pores, like the what can I say, like a cellular pores. Cellular pores are there, so it is necessity to absorption of this drug. Molecular weight is very important parameters, because molecular weight can if the molecular weight is high, it cannot be incorporated in the body. If permeability will be low, then will come to a lipophilicity. Lipophilicity is a log p value. It is determined by the lipophilicity. Log p value is actually the drug. What is that drug is incorporated in by orally or intravenous. It will be mm, it will be in a manner that octanol. You know about octanol. Octanol is a compound that make it is a lipophilic compound, and water is the another compound. So log p value actually denotes that octanol by log of octanol by water absorbance. So lipophilicity going through this. It then comes to the hydrogen bond donors and acceptors. And hydrogen bond donors and acceptors means that these phytochemical properties goes through the chemically synthesizing property. Like what if hydrogen bond donors are top, more, sorry, if it is too much, if the hydrogen donors are too much and acceptors are too much, what will happen? It will affect the chemical properties of the drug, and it can affect the toxicity. So it is the main topic. Then we can come to the topological polar surface area. Actually, polar surface area is denoted as the surface area of the drug that is acquired by the polar compounds. Polar compounds means that have a ionized, because it is ionization is very needed a parameter, because lipophilicity depends on ionization. So that topological polar surface area is a very important key physiochemical property. Then rotable bonds. Rotatable bonds, it is a class 12. Can I question if something that what you know about the rotatable bonds? Like I am asking you that a class 12 chemistry all know that there are the chiral bonds. So if it is going through the chiral bonds, if one chiral bond property is there in a chemical structure, what will be happen in that? Can you say?
Adhikari, actually, one participant has raised their hand, Adi Punusami.
Okay, okay.
Adi Punusami, you want to, you can unmute and you can question anything if you want.
Yes, yes. Adi, can you question me?
Either you can chat, or you can message, or you can unmute and ask, or else he can continue. Anything, Adi?
Adi?
If nothing, I think Dikari, you can continue.
Okay, sir. I am questioning that uh, if a chiral bond is there in a drug, what problem comes through uh, the absorption and the other properties? Can you explain? Yes, very good. Naveen Suresh, you have pointed out a very important property, that is isomerism. Isomerism, you know about the isomerism, isomerism is a property that can be convert a chemical property. It is a chemical property that isomer is a type of what can I say, that like a chemical compound has different chemical structures and is shifted their chemical structures by moving their chiral bonds. So isomerism property is the main problem in this thing, because when we it permeable permeable to the plasma membrane, that time it's come to the lipophilicity is the comes to then the main part, like that. If it comes and it rotatable bonds are there is too high, then it cannot pass through the plasma membrane, because there will be some hindrance. If if the structure is very rigid, the rotatable bond numbers is very less, then it can move easily through the plasma membrane. So that this property is very important property for that. So, and lastly, the solubility. Solubility, you know that soluble in lipid or soluble in water. If soluble in water, that it is the lipophilicity is very low, and this is soluble in lipid, the lipophilicity will be high. So log p value differs between the solubility. Let's come to the next point. Drug likeness properties. Lipinski rule of 5 is also.
Share your presentation. It's
Oh,
not
sir,
in
sir, sir.
presentation.
Okay, okay, okay. Now you can see, sir.
Yes, yes.
Yes, sir. Drug likeness properties. So we are coming to the Lipinski rule of 5. It is also known as Caesar's rule of 5 or rule of 5. It's not about that it is the 5 rules, but it has the 4 rules actually. Actually, it has the 4 rules, but it comes in the actually it comes in the time of what I can say. This is rule of thumb. You can say it like there is there is actual 4 rules, but in after that incorporation it is comes to the 5. Like molecular weight will be less than 500 gram per mole of a drug. Log P will be less than 5. Hydrogen bond acceptor and hydrogen bond donor will be hydrogen bond acceptor will be less than 10, and hydrogen bond donor will be less than 5. And the PSA, I have told you that PSA, the polar surface uh, uh, area, polar surface area is is comes to the 140 angstrom, less than 140 angstrom, less than 140 angstrom, and RB value, the rotatable bond is less than 10. So these 5 rules are coming in that by Lipinski, set by the Lipinski, and this is the rule of 5. This rule of 5 elevate the drug likeness or determine if a chemical compound with certain pharmacological or biological activity as a chemical property and a physical property that would make it a likely orally active in drug in humans. Like that, if you can say that what is bioavailability, it comes in that in this figure. Bioavailability means how much of the drug actually enters in the bloodstream and it is available to exert its therapeutic effect. By means, what is that? Like if you take a drug, if it is entering in your body, how much percentage is going to your blood? That is main thing in this terms. Okay, can you understand? Actually, the blood in the the in the form of oral liberation, how much drug we takes, it not take properly to the it not comes the whole drug comes to the blood, because there are some uh, ADME properties. ADME properties, what is this? I am coming to the ADME: liberation, absorption, metabolism, and excretion. When the absorption is the blood of it is a abbreviation. It is an abbreviation of a pharmacokinetics and pharmacology for the absorption, distribution, metabolism, and excretion. So you know about the admin, but what how it is come into the this state? Actually, bioavailability is the main concept of the admin. How much of the drug actually enters in the blood. This after absorption, metabolism, absorption, distribution, metabolism, and excretion, the percentage of the percentage of the drug, it comes very low, and it comes to the blood. It's very low amount. So we have to increase this amount via this drug likeliness property. Like, if bioavailability suddenly increase due to liver disease or any drug interaction, the the drug will concentration may be rise in the blood, which directly incorporate toxicity. Like that,
Uh,
yes,
Adhikari, so can I take a lead? Can I go ahead with my can I do this presentation?
yes, sir, yes, sir.
Okay. So you can stop your presentation. I can start my presentation if you don't mind.
Okay, sir.
Now I am sharing my presentation. Can I?
Yes, sir.
Okay. Thank you, Adhikari. So very good. You have taken a good lead. Now I just continue with wherever we just stopped. Okay, terminated. So you are explaining very good about the drug likeness prediction. Let me complete it because we have a very less time for even to show the hands-on session. So I thank Adhikari for to mention something about drug likeness and ADME. So let me go ahead further. So he was mentioning about the Lipinski rule of five. Okay, so the Lipinski is one of the drug likeness filter. This also called as Pfizer's rule of five. Okay, so if you see this, why we call this as rule of five, because the properties of drug, okay, properties of drug ends with multiples of five. So if you see the Lipinski rule of five, there are actually four properties. The first property what we can take is the molecular weight, which should be less than 500 gram per mole. And the second property is partition coefficient. We measure in log P, which should be less than 5. And similarly, the third property is hydrogen bond acceptor, and the fourth property is hydrogen bond donor, which should be less than 10 and less than 5. So according to Lipinski rule of 5, if a compound, like if you take a compound extracted from a plant, that's what he explained, Adhikari, like a mangiferin. Mangiferin is a plant compound from which is obtained from Magnifera indica leaves. Okay, this is used for treatment of heart problems, cardiac problems. Okay, like if you want to check this plant compound is proving to be a drug or not, which can shows a good kind of a oral bioavailability or not. So to check this, we can use this kind of a drug likeness filter, like a Lipinski rule of five. Many scientists adhere to this kind of drug likeness filter. According to this Lipinski rule of five, okay, out of these four properties, if any three properties are satisfying out of four, we can call that as a that compound as a drug. So if mangiferin shows all the four properties, okay, if the properties of the plant compound shows all these satisfy all these rule, then obviously we can still term it as a drug. Okay, so if you see the additional properties available, rotational bonds and polar surface area, these are present in other drug likeness filters. Lipinski rule of five is one drug likeness filter. Like this, we have other drug likeness filters. Okay, and I think he is. I'll be taking you to the other drug likeness filter directly to the website. Now, let me take you to the uh, information on ADMET. So now I am taking you to the website called IMPAT. So IMPAT, this is the largest Indian medicinal plants phytochemistry and therapeutic database. Here you find information on drug likeness, ADMET properties, descriptors, and we also can find the predicted human target proteins. Okay, so like however I mentioned, like Pfizer rule, we have GSK rule, Egan rule, Weber rule, Goose rule. These rules are the other drug likeness filters. So they have their own kind of a property values. Like however we have a threshold of molecular weight less than 500, we have a different threshold for the Weber, Goose, Egan, etc. Okay, and now let me take you to the next website, that is Herb 2.0. This is a traditional Chinese medicine database. This I have already shown you in the session 2. Okay, here why I showed you is here also I can able to find the drug likeness filter values. So the drug likeness filter they have used a correlation with all the physicochemical properties of the drug, and they've estimated a value. The value they estimated for a plant compound, the plant compound I have given as an input is a vitafrin A. For that I got 0.485. Okay, correlation was developed with these properties, whatever Radhika has mentioned, molecular weight, polar surface area. Okay, so these drug likeness filters, or you, I mean, this physicochemical properties of the drug is taken to calculate the drug likeness. Now, let me take you to the output of drug likeness from a third software called MolSoft. MolSoft is an online tool where it requires a kind of a input of smiles format of a compound. Here in this case, I have given an input of curcumin. Curcumin, I think okay, this is a compound from curcuma longa. So if you provide a smiles format, canonical smiles format, okay, obviously you can able to see this physicochemical properties of the compound. So from this physicochemical properties, we can check whether this can act like a drug or not using even Lipinski rule. For example, if you see the molecular weight as according to the I take the eraser. So according to the Lipinski rule of 5, what you can able to understand, if it is less than 500, it proves like a drug, isn't it? The compound shows less than 500, obviously here, and the hydrogen bond acceptor should be less than 10. In our case, it's 6, so it's obviously it is less than 10. And according to the Lipinski rule of 5, the donut should be less than 5. So already the three properties are satisfied. The fourth property, if you remember, the log p value. The log p value here in this case is 5, and we should get less than 5. Obviously, if you check all these four properties for this curcumin compound is satisfying, so we can tell this compound is satisfying the Lipinski's rule of 5. So this compound is behaves like a drug. So now we can tell this is working like a drug. And also you can see, according to the MolSoft, you also get a value here in the bottom. The bottom line is streak line, a red line you can able to find, isn't it? So from this you can able to find the drug likeness score. Okay, if the drug likeness score should be between minus 1.5 to plus 1.5. So just because our value is it's not extending minus 1.5. So even in the graph it is showing, so this is the value actually minus 0.82. So minus 0.82 is lying between minus 1.5 to plus 1.5. So again, this graph also shows from the MolSoft that our compound behaves like a drug. So like this, you can even check with the online tools. There is another tool called Mol Inspiration. In the Mol Inspiration, still you can check the same, like how I checked with the MolSoft. Now let me take you to the ADMET. ADMET, you can also include one more thing called L. L stands for liberation. As I earlier mentioned, like even Adhikari has mentioned that for the drug likeness, okay, we check initially whether a compound behaves like a drug or not. The compound if you take from a phytochemical source, like example mangiferin, okay, how we prefer to take this kind of a compound drug in the form of a oral route. Even a kid or elder people, if you want to give a drug, whether they like to take in the form of injection, no, isn't it? Many of them don't like to take a drug in the form of an injection, intravenous injections, so infusions. So if this route is not, this drug delivery technique is not preferred, obviously many try to choose only the oral route, isn't it? So if you take it to the oral bioavailability, whatever drug taken in the buccal cavity, the drug is going to transfer from the buccal cavity to the absorption. The second process is the absorption. The absorption is generally is going to happen in the blood. Okay, absorption is going to happen in the blood, and the distribution is going to happen in the tissues, and metabolism is going to happen in the liver, where you can able to find the fast pass metabolism. Okay, that is any drug. Okay, if you are taking in the oral route, 50% of the drug, or even more than the 50% of the drug, going to metabolize in the liver. The reason why it is going to metabolize in the liver is we have so many cytochrome P450 enzymes. The cytochrome P450 enzymes present in the liver try to metabolize any kind of a drug, lipophilic or hydrophilic drugs. Okay, then to avoid this, obviously, we take a different kind of a drug delivery, drug route technique. We take even injections, intramuscular, intravenous, etc. But okay, if, for example, if we take a oral route, obviously, we have to even check this kind of a liver, okay, where we can find major kind of a metabolism is going to happen. And this kind of a metabolism, we call it as a first pass metabolism, okay, which happens through the cytochrome P450 enzymes. If you want to escape from the decay of much of the drug in liver, obviously we have to improvise the drug delivery technique in the form of liposome, neosome. There are so many kind of drug delivery techniques, isn't it? We can encapsulate drug with lipids so that it's not going to decay much in the first pass metabolism in liver. Okay, so then the next is the excretion. E stands for excretion or elimination. Okay, the unutilized drug, whatever the drug is not going to bound with the plasma protein, is going to excrete in the form of urine, feces, or even the sweat. Okay, this is how the drug is going to eliminate. So, this is what we call it as LADME. Then what is this T? Can someone tell me what is T? I think we are already discussed on session 2. Yeah, what is T?
So toxicity.
Toxicity.
Exactly, exactly. The toxicity, we have different types of toxicity: nephrotoxicity, neurotoxicity, cardiotoxicity, okay, hepatotoxicity. So many toxicity are available. Okay, so one should also have to check whether your drug is going to have a toxicity or not. Okay, so we have different online tools and databases to check this kind of an LADMET properties. So the popular softwares what we use is a Swiss ADME, and few of the tools or commercial tools. Okay, but we have free tools to work, so why we have to worry about the commercial part? Now let me take you to the Swiss ADME. In Swiss, this is an output of a Swiss ADME. Once again, I have given input of curcumin. So if you see this curcumin, okay, so whatever I feed a smiles format, a canonical smiles format is an input for this tool. So if I feed this canonical smiles format, we can able to find this kind of a properties like molecular weight. Again, well, I've already checked in the MolSoft tool, isn't it? So no need to check all this. I just showing you we have a physicochemical properties. Okay, other than physicochemical properties, we have lipophilicity properties. We can able to check the partition coefficient values. We have types of partition coefficient. So as clearly explained by Adhikari, okay, that is the partition coefficient we can able to measure through octanol water partition coefficient. Actually, whatever we try to check the effect of drug in our body, we can also check using this chemical solvents. Octanol water. Do you know why we use this kind of an octanol water experiment to check the partition coefficient? No, the reason is, if you know the answer, you can answer. I can wait for 5 to 10 seconds. Later, I'll be providing it. Why we have
Sir,
to use?
because it resembles the nature of the body, sir, when it comes to lipophilicity and hydrophilicity.
Very good. Good answer. Can I give you still more clear answer? If your answer is right, I can give you an still more clarity that the pH of the octanol is 7.4. Did you hear about the pH 7.4? Which part of the body has pH 7.4?
So blood.
Exactly. Okay. So the lipophilicity we are linking with blood. And I already told you in yesterday that more than 70% of the body is made up of water, is comprises of water. So lipophilicity versus hydrophilicity. For lipophilicity, we take blood, and for hydrophilicity, we take water in our body. Okay, so pH 7.4, and we check with the. So this is what we choose even the log part B for checking the lipophilicity and hydrophilicity. Okay, now let me take you to the water solubility. Even this is something linked with both. If this is lipophilicity, this is hydrophilicity, no doubt in that. So hydrophilicity is opposite to hydrophobicity, and lipophilicity is opposite to uh, okay. Lipophilicity is hydrophobicity, and hydrophilicity is lipophobicity. Pharmacokinetics. In pharmacokinetics, okay, I told you we can able to check ADME properties. So absorption can be determined using gastrointestinal absorption. So the curcumin, you can see the high absorption in the stomach, and the BBB stands for distribution. That is, you know, BBB stands for blood-brain barrier. Blood-brain barrier. So why we discuss like this? Even the blood. If you compare brain, blood is having less lipophilicity. Our brain is having more lipophilicity. So if your drug has to be penetrate to the brain, that is, if when we go across these kind of a property for a drug, if you want to use a drug to treat central nervous disorders, peripheral nervous disorders, so majorly your target organ is the brain or spinal cord, which is more lipophilic. If only if a drug is more lipophilic, then only the drug is going to penetrate the brain. Then only you can able to treat the brain disorders. Got it? So this blood-brain barrier permeant denotes the more lipophilic, and of course that is what we denote for distribution. Distribution means distribution to the tissue. For example, if you want to treat Alzheimer's disease, Parkinson disease, we have to distribute the drug only to the brain or spinal. That's the most important for several central nervous disorders. So, distributions we can check with the blood-brain barrier permeant. If you are using curcumin for treatment of cold, cough, why there is a need to reach the brain? So if you get no, this is a desired property. If you get GI absorption for the cold and cough, still this is a preferred property. And similarly, we can go with the metabolism. The metabolism we can able to check with the cytochrome P450 enzymes. If you want to know about the enzymes, you can click this question mark. Then you can find more information of this cytochrome P450. Then you can check whether yes and no or desired or not. And finally, E stand for the excretion. The excretion, that is unutilized drug is one is going to excrete, isn't it? I told you even the excretion happen in the form of sweat. Then skin permeation is important. The unutilized drug is going to excrete in the form of sweat, and this is the value for this kind of a drug. You got it? And if you want to check with the drug likeness, okay, we have different drug likeness filters. I told you earlier, Lipinski. So the curcumin, I already proved in the MolSoft that is having no violation, zero violation. So the drug likeness is yes. And even Goose, Weber, Egan, we have different values for the molecular weight, log partition coefficient, etc. If you want to know more about this, click this, go to the Swiss ADME and click this question mark. You will be getting the properties of it. I'll be showing you when I take you to the browser. Now let me take you to the pre-admit. This is another tool. So earlier I've gone to the Swiss ADME. Now we are going to the pre-ADMET. In addition to the ADME properties, whatever we checked in Swiss ADME, we also have T, toxicity prediction. The toxicity prediction we are predicting using the Ames test and rodent carcinogenicity assay. Okay, so these are the additional features in this pre-admit. Of course, it's an online tool. One thing is that you need a kind of a smiles format. The smiles format is the input. You can feed for a chemical compound and you can check the admit properties using pre-ADMET. There is also one more tool called PKCSM admit. So using this also we can predict. Okay, one is smiles format, you have to input and you have to You can check absorption, distribution, metabolism, excretion, toxicity. However, you find the output in the pre-admit, the same kind of output will be reflected here for the same compound. And more about the absorption, distribution, metabolism, the values, everything, the output page is shown here. So first try with any kind of a compound like a curcumin and check these kind of outputs. So for Swiss target prediction, this is another tool, okay, which is available in the same kind Swiss server, where here it just ask you for an input of smiles format, give an input, okay, and click like predict targets. You'll be getting a targets like this. Do you know what is this? This is of course a kind of a target deconvolution. You remember on the first day I told you target deconvolution. So I missed it. Okay, now I am showing you this deconvolution. Okay, so you can see there's a series of receptors and whether our compound, the compound what I input is withanolide A. So one of the plant compound is steroidal lactone compound from ashwagandha. When I input this, I can able to find this compound is going to target what type of receptors, what classification of receptor. Like this, you can try with any compounds. And you can also check a series of receptors and how about the binding capacity. Okay, so this is about Swiss target prediction. Other than Swiss ADME, I'm showing you another tool for their target deconvolution. Now let me take you to the another tool called Protox. Now this is of course a most important tool for predicting the toxicity. You remember on yesterday I explained you on drug-drug interactions, drug-food interactions. Of course, in the drug-drug interaction and drug-food interaction of drug bank, we are also checking the adverse effects, side effects, of course related to toxicity. But toxicity is something more than the side effect and adverse effects. If you want to use a drug, a particular concentration of a drug, to check the activity of a drug in cell line, for example, cancer cell line, in vitro studies, or if you want to check the concentration of a drug to be tested in vivo experiments, in mice, in rat, whatever drug you take for testing in mice, you can't able to use the same concentration for rat. And whatever drug concentration we test in mice and rat, we can't able to take it, use it for human. So we have to know the toxicity in each and every animal. Okay, the toxicity is pertaining to a specific species. Okay, here whatever toxicity they mention. Okay, so we have space. We are checking with respect to human. Okay, if it is mice, we are checking with mice. If you see this earlier slide, I showed you like here whatever toxicity is predicted is for the rodent. The rodent, like mostly here they have given for rat. Okay, like this you have to check for the tools, what tool is which tool and what species. That's also most important. If even if you see the Swiss target prediction, you can see whether you are checking for homo sapiens or is checking for mus musculus or ratus norvegicus. Don't predict as such for that whatever we predict for human can be used with mus and that is mice and rat. So and whatever you predict for mice can be used for human as such. Okay, so you be specific even on the species what exactly you are searching for. You are searching for toxicity or even searching for any kind of adverse and side effects. Okay, carefully tracing the tools is needed if you're working on drug design and discovery. Now let me take you to this toxicity prediction. I told you Protox 3.0 is the recent version of the server to predict the toxicity. All the types of toxicity can be predicted in this toxicity prediction. First you have to click this. Okay, and if you click this, you obviously get an input to input a canonical smiles format. Here you have to give a canonical smiles format, or you just give the pubchem name. So obviously you can get a compound and simply go down and click submit. So this is the home page. Actually in the home page you have to click here and you have to go inside and try to predict the compound toxicity. So if you click there, obviously this is the window you get. This is an input window. After giving feeding a smiles format of the compound, what toxicity you want to predict? Whether you want to predict organ toxicity. If I am checking for hepatocellular carcinoma, if I want to check my drug is going to work well for the liver cancer, obviously I have to check for hepatotoxicity. But whatever drugs we use for the liver cancer or breast cancer or whatever type of cancer, part of the drug is utilized or is distributed, but part of the drug is not going to unutilized, not going to distribute to the tissues, going to excrete. If the some part of the drug is not going to utilize and going to excrete, obviously we have to check with the nephrotoxicity, because the major unutilized drug is excreted only through kidney, isn't it? That's the most important organ. Okay, not only it's regulating the blood glucose, but the unutilized drug is going to excrete in the through the kidney. So we have to check obviously the nephrotoxicity. And if you are working on specifically lung cancer, instead of hepatotoxicity, go with the respiratory toxicity. So like this, you can check with what toxicity you want, and if you want all the types of toxicity to be checked, you can check all this. And similarly, you can check with the toxicity endpoints, and we can check with the nuclear receptor signaling pathways. We have stress response pathways. We have molecular initiating events, and we have metabolism, cytochrome P450 enzymes. So all these are part of a toxicity. To check all this toxicity in terms of signaling pathways, metabolism, we can able to check it. I think I have already shared you these kind of in videos related to plant-derived secondary metabolites for the treatment of diseases to a company. So also check this, which might be helpful for your project. Now let me take you to the practical hands-on session. So till now we have discussed the theoretical part. Can I take you to the hands-on session at present? Is that okay? Can I go ahead? Am I audible to everyone? Just kindly interact with me.
Yes, sir.
Good,
Yes, sir.
thank you. Now let me go ahead. So first I'll be taking you to the Swiss ADME. Before going to the Swiss ADME, we have to take a input format from Pumpkin. So Pumpkin, you find a 1D, 2D, and 3D structures of chemical compounds. For example, if you want to work with curcumin, or if you know any compound, or mangiferin, you mentioned, isn't it? Mangiferin. Okay, this is the compound we are going to try now. Already we checked with curcumin. Now we are going to check with the mangiferin. Let us go to the mangiferin. So mangiferin is a compound from Mangiferin indica. From mango leaves, you can able to get this compound. So if you want to get a smiles format, okay, we also have a 2D format. If you want to work with the 2D format, generally we won't prefer to go download as 2D format. We prefer to download a 3D format. Okay, in vaccine design also we use the 3D format of the peptides. So if you want to use a 3D format, I suggest you to download the 3D format like SDF, structured data format. So if you want to download the 3D, we can download this. But in our case, we need only the smiles format. So drag down and check for the smiles, which is nothing but 1D format. Whatever I'm telling like a smiles, canonical smiles is a 1D format of a chemical compound. This is the 1D format. I just copied it, but let me take you to the tool. The tool, whatever I'm going to illustrate you at present, is the Swiss ADME. So go to the Swiss ADME. Now let me take you to the prediction of drug likeness properties as well as ADME properties in this tool. So only thing is that you have to paste the smiles format here. I pasted it. Simply click run. So if I click run, you'll be getting the result instantly. No need to wait for up to 5 to 10 minutes like Swiss modeler. You'll be getting the result soon. See, the results are generated at present. Now let me show you what is the result we get. The first result what we get is the show boiled egg. If I click this show boiled egg, okay, you can able to see this egg. Okay, that is white albumin protein with yolk sac. Where is my compound? The compound is not present everywhere. Actually, it's getting loaded. We don't find the compound. We'll be getting the compound somewhere soon. White boiled egg, show. I have to refresh it. If I refresh it, I can able to see this blue color kind of a dot. So now let me take you further. What are the properties we can able to see here? The molecular weight 422, which is less than 500, and the number of hydrogen bond acceptor should be less than 10. See, we are violating the Lipinski rule is violating here. One violation we already found. Donut should be less than 5. Again, second violation is found. So that's what they have shown here. In Lipinski rule, two violations are found. How about partition coefficient? Partition coefficient is 0.89, less than 5. So partition coefficient is no violation. Molecular weight also we don't find any violation. And similarly, if we check with the goose filter, these are the properties. Molecular weight should be 160 to 480. Log partition coefficient should be minus 0.4 to 5.6. See, the properties are varying. Okay, and similarly, if you see the Weber, there are actually two properties, rotable bonds and total polar surface area. The rotable bond should be less than or equals 10 and total polar surface area less than or equals 140. Got it? So like this, you can see a different physicochemical properties of the drug mentioned in this drug likeness filter. You can also see the GA absorption like ADME properties from the pharmacokinetic property. So this is something about how to use the Swiss ADME. Okay. Sometimes if your internet is slow, you can't able to find the blue dot. For example, if my drug is lying in this albumin protein, okay, if my drug is lying in between, I mean in this white protein, then you can tell this is a lipophilic drug. No, sorry, hydrophilic drug. This is a hydrophilic drug. But if my drug is lying in this yolk sac, then I can tell my drug is lipophilic drug. Yolk is more lipophilic, and the white albumin protein is hydrophilic. This is how we can able to analyze from the boiled egg diagram. Now let me take you to the Swiss target prediction. So just I completed Swiss ADME. I just need 2 to 3 minutes to complete the prediction. Let me take you further on Swiss target prediction. This is related to target deconvolution. I told you even if I miss anything in the previous sessions, if I have a time, I can able to take it further. Okay. So see here also, I think server is slow. What to do? I can't able to predict target. But if my server is fast, okay, it depends on the server. Again, I'm refreshing it. If our server is fast, then obviously we can check for the different species. Examples, or I just give triclosan, still predicts. Provide a smiles before submitting. See, I've given the example, but still it's not selecting. So the reason behind is the server is too slow. So better we can check it on some other day. If we check it on some other day, we'll be getting an answer. No doubt in that. Now let me take you to the next tool, pre-admit. So how to work with pre-admit? So pre-admit, we have a tool here. So in Google, if you search with the pre-admit, we are getting this tool. So if I want to check the toxicity directly, we already checked ADME from Swiss ADME, isn't it? If I want to take check the toxicity, they're asking you to draw the molecule, okay? Or if even the input is, if they ask an input, you can feed the input. So you can draw a molecule, and you can predict the. For example, I'm going to draw a molecule. So there's a peak load. So the problem is with the server. So simply you have to draw a molecule. Am I audible to you? I don't know.
Yes, sir. Yes,
Yes,
yes.
yes. So we have to either provide a smiles format, or you can draw a molecule and you can predict it. I just selected and clicked. We have to get a structure. Meanwhile, can I take you to the Protox 3? Protox 3. So this is also used for toxicity prediction, isn't it? Let me take you to the Protox 3. So to predict the toxicity prediction, canonical smiles, paste any canonical smiles. Click this smiles. You got a kind of a compound. Whatever compound we got to predict the toxicity, okay, in Protox 3, you have to select. If I want to check hepatotoxicity, nephrotoxicity, I'm selecting this. And if I want to select any specific mitochondria, whether this have a mitochondrial membrane potential, I am selecting this. Okay, and then if I click start toxicity prediction, you'll be getting the result for that. You know, the predicted LD lethal dose 50 is 2 milligram per kg. Is 2 milligram per kg. I think presently it has taken a rat, I believe. Okay, so for this species, this is the lethal dose. Okay, And if you see, the toxicity is very, very less because 2 mg is very less, isn't it? And if you, I mean, the predicted toxicity is 2 milligram per kg. Now, related to properties, you can find. Now, let me check with the organ toxicity. If you check the organ toxicity, hepatotoxicity is inactive. So, this is not going to give any toxic to the liver, but it is going to give toxic to the nephrons, that is kidney. Okay, whatever drug I pasted, it is going to give a nephrotoxicity. And mitochondrial membrane potential, whether this drug is going to penetrate in the mitochondria or not, whether this can be used as a mitochondrial targeting drug or not. No, we can't able to use. So inactive. It means that it's not going to penetrate the mitochondria. This is not a mitochondrial targeting drug. So like this, we can able to infer the results using this toxicity. That is Protox 3 for analyzing the toxicity. Pre-admit, still the servers are too slow. So if you work on early morning, you may get a kind of a result. Okay, it's not linked with our internet frequency. This is linked with the server server loading capacity. Okay, so even on tomorrow, if possible, we can check, or you can check even in your phone. It works perfectly. You just give a compound structure, draw a structure, and you have to simply click. Submit to check the toxicity. Okay, so today I've completed about drug likeness and ADMT properties. Any doubts you can ask me at present. Do you have any doubts? So if no doubts, thank you very much for your interaction. Okay, so tomorrow we'll be continuing for day 4 session. Okay, so have a great day to you all.